Hello students, this is Shayista from Microbiology Department KSOU. Today I am dealing with the topic factors affecting enzyme activity. To begin with, let me give you a brief introduction. The cells are amazing molecular machines. Every day they convert food into energy, which we call as ATP, adenosine triphosphate. These ATPs in turn drives many biochemical processes for keeping the cell alive. The pathways used to convert substrate like glucose into products are collectively called as metabolic pathways. Here in this image, we can see many metabolic pathways like glycolysis, which converts glucose into pyruvic acid, and in turn this pyruvic acid is used as substrate in Krebs cycle, that is our TCA cycle. And simultaneously the electron transport chain together producing ATPs. That's our energy. The drivers of these metabolic pathways are the enzymes. Every biological pathway has its own set of enzymes which are highly specific in nature. So what are enzymes? Enzymes are biocatalysts. They are usually proteins that work together to assist chemical reactions by either building or breaking down the molecules. Enzymes speed up the rate of chemical reaction in which they participate and do not get destroyed and can be used over and over again indefinitely. But the rate of a reaction is never constant. Various factors play an important role in its regulation. It can either speed up the reaction or slow down a reaction or completely stop the reaction. And this combined together is called as the regulation of enzymes. What are the factors affecting the enzyme activity? The factors which affect the enzyme activity are many, but basically it can be the concentration of the enzyme, the concentration of the substrate present, the effect of the temperature and the pH, covalent modification occurring because of the presence of some other biological entity, and the inhibitors. The inhibitors are basically of two types. It can be either a reversible inhibition or a irreversible inhibition. And in reversible inhibition, we have again two types. It is competitive inhibitor or a non-competitive inhibitor. When a question is asked about what are the factors affecting an enzyme activity, all these factors must be taken into a consideration. In this class, we will be dealing with each subunit in detail. Now let's begin with our first subtype, that is the effect of enzyme concentration on enzyme activity. Here in the graph, it is plotted with rate of reaction on y-axis and enzyme concentration on x-axis. Let's take up a condition where the substrate is unlimited. That is, the substrate is in constant supply. Initially, as the enzyme concentration increases, the rate of reaction also increases. It is clearly shown in the graph with the straight line. That means the rate of reaction is directly proportional to the enzyme concentration. The condition here taken is that the substrate is unlimited. But when there is a second condition that the substrate is limited. When the substrate is limitedly present in the system and the enzyme concentration increases, the rate of reaction increases with increasing in the enzyme concentration. But there comes the time when enzyme concentration is more than the substrate concentration and the graph levels out. The rate of reaction saturates or decreases because of the collision between the enzymes and the substrate or the enzymes or the product. Here, the rate of reaction will be independent of the enzyme concentration increasing because the substrate is limited. Now, let's deal with the effect of substrate concentration on the enzyme activity. So, a similar graph is plotted here again with a rate of reaction on y-axis and the substrate concentration on the x-axis. The rate of an enzyme catalyzed reaction is always affected by substrate concentration. As the substrate concentration increases, the rate of reaction also increases because more substrate molecules can collide with active sites. Therefore, more enzyme substrate complexes are formed. At higher concentration, the enzyme molecules become saturated with substrate and there are very few active sites available. So, adding a substrate will not make any difference in the further rate of reaction. Here the condition is taken that the concentration of the enzyme is fixed. As explained in the graph, you can see the rate of reaction increases with the increase in the substrate concentration. But when the substrate concentration is greater than the readily available sites, active sites on the enzyme, the rate of reactions will saturate because all the active sites on the enzyme will be occupied. And the enzyme is in fixed concentration. You cannot add any enzyme. And thus, this graph shows the effect of substrate concentration on the enzyme activity. Further explaining the effect of substrate concentration on the enzyme activity, Michaelis-Menten kinetics plot helps us to give us a clear picture. This graph is similar to the earlier graph which we saw, that the increase in the concentration initially increases the rate of reaction, and as the concentration of the substrate increases, the reaction rate levels out because of the non-availability of the enzymes. At higher concentration of enzymes, the enzyme molecules become saturated with the substrate. There is no free active sites left to engage the substrate. Thus, the graph is leveling out. In this graph, you can see is Vmax. Here, the Vmax is the maximum velocity or the rate of reaction at which the reaction is occurring. And this can be independent of the substrate concentration after the Vmax is achieved. Thus, Vmax can be said as the maximum rate of reaction at infinite concentration of the substrate. Now, here we can see what is half Vmax. Half Vmax is the more efficient velocity of the enzyme. Michaelis-Menten said that half Vmax, that is half Vmax divided by 2, half the value of Vmax is the efficient velocity. That is the the work of an enzyme can be done at this velocity. So the Km value, the Km is nothing but the amount of substrate that is used or the amount of concentration of the substrate which gives us the half Vmax value. So why are we seeing this equation? This equation helps us to characterize a good enzyme. According to Michaelis-Menten, a good enzyme will always have a high Vmax value and a low Km value. What do you mean by low Km value here? Low Km value is if the value has to be low Km, then the amount of substrate concentration to reach an efficient velocity of a reaction or the rate of reaction has to be low. That is, the concentration of the enzyme should be least so that soon half Vmax value can be achieved. Thus, a good enzyme must always have a high Vmax value and a low Km value. Now, students, let's study the effect of temperature on enzyme activity. Similarly, a graph is taken here where on y-axis the rate of reaction is plotted and on x-axis the temperature increase in the temperature is pointed. So let's see at this graph. So initially you can see that the rate of enzyme reaction is very low. Why? Because of the low temperature. At low temperature it is very difficult for an enzyme to function. And there is a steady increase in the rate of reaction. This increase is not directly proportional. As you can see, this line is not a straight line, it is slightly curved line. So there is a steady increase in the rate of reaction with increase in the temperature until a optimum temperature is reached. When a optimum temperature is reached, the rate of reaction of that specific enzyme is highest. So every enzyme has its specific optimum temperature. There is no fixed temperature that at this universal temperature all enzyme will function. No, there is no such condition because every enzyme will have its own optimum specific temperature. Now, after this, when the temperature increases after the optimum temperature, you can see a sudden sharp decrease or decline in the rate of activity of the enzyme. There is a sudden sharp decrease. This is because the high temperature will cause denaturation of the proteins. And as we all know, the enzymes are made up of proteins. The thermal energy breaks down the hydrogen bonds that holds the secondary and tertiary structure of the enzyme together. So the enzyme loses its shape and becomes a random coil, and thus the substrate cannot fit into the active site and the enzyme is permanently denatured, and this reaction is irreversible. So students, let us now study the effect of pH on enzyme activity. Enzymes have optimum pH at which they work fastest. Just like we studied earlier that every enzyme has a specific optimum temperature at which they work fastest, same way every enzyme has a specific optimum pH at which they work fastest. For most enzymes, this specific optimum pH ranges from 7 to 8, which is normal body pH. But for few enzymes, they have extreme pH. For example, let us consider gastric protease. The enzyme which is commonly known as pepsin that is present in our stomach has an optimum pH of 1. So in this graph, you can see there is a comparative study between different enzymes, that is pepsin, trypsin, alkaline phosphatase. 3 different enzymes are taken and there is a comparative study done of the effect of pH and the activity. So now, as I said earlier, the pepsin has its optimum high activity at an optimum pH of 1. Similarly, trypsin has its high activity at its optimum pH of approximately 7, and alkaline phosphatase has its maximum activity at its optimum pH of 10. So by this graph, we can conclude that every enzyme has an optimum pH, and this pH, it works faster. Rising or lowering the pH can cause change in acidic and basic side chains of enzyme, thus they may denature. So now when I am altering the pH, there is denaturing of the protein occurring. Therefore, there is a steady sharp decline in the activity of the enzyme. The pH affects the charge of amino acid present in the active site. So when the charge of amino acid is changed, the property of the active site is also changed. Therefore, no further substrate can bind. When the pH is altered, the active site property changes, thus inhibiting the further binding of the substrate. Thus, the activity completely stops. Another factor which affects the enzyme activity is covalent modifications. Now, these covalent modifications can occur in enzyme because it can be controlled by some other enzymes that is present in the system. And how are these covalent modification occurring? These modification occur either by cutting the enzyme or the other enzyme adding a phosphate group on the enzyme or by adding a methyl group. All these clubbed together can either turn the enzyme active or inactive. The best example that can be given is the HCl, which is released in our stomachs, activates the enzyme pepsin, which in turn activates the enzyme renin. This can be an perfect example of an biological entity which allows covalent modification occurring in other enzymes so that they can turn active and further function in the biological systems. The enzyme activities is further affected by a substance called inhibitors. An enzyme inhibitor is a substance that decreases the rate of reaction of an enzyme catalyzed reaction. So basically, we have two types of inhibitors: reversible inhibitors or irreversible inhibitors. Inhibitors inhibit the activity of enzyme, reducing the rate of reaction. They are actually found naturally or can be used artificially as drugs. Naturally, they are found in cells. They inhibit specific enzymes, providing a means for internal regulation of cell metabolism. And artificially, they may include medicines or many poisons which inhibit one or more enzymes and thus decrease the rate of reactions. So now we have a comparative study about these inhibitors. They are of two types. It is either a reversible inhibitor or a irreversible inhibitor. Here the reversible inhibitor binds with a non-covalent interaction, but whereas the irreversible inhibitor binds with a covalent interaction. They do not perform any chemical change in reversible inhibitors. But whereas an inhibitor which is irreversible, it binds to the substrate and prevents the catalytic activity of the enzyme, thus bringing about chemical changes both in the substrate as well as in the enzyme. The reversible inhibitor can be reversed as there is no bonding between the inhibitor and the substrate. And moreover, it is non-covalent interaction which can be easily broken down. But in irreversible, the reaction is very strong because of the covalent bonding, and thus it is not reversible. So students, let us now focus on types of reversible inhibitors. There are two types of reversible inhibitors. One is competitive inhibitors and the second one is non-competitive inhibitors. In competitive inhibitors, let's first look at this diagram. In this diagram, the substrate binds to the enzyme active site, results in the formation of enzyme substrate complex, which further proceeds in the forming of the products. Now, when we consider a competitive inhibitor, this competitive inhibitor has a structural feature similar to that of substrate, so that it can fit itself into the active sites of the enzyme. And when it gets fit into the active sites of the enzyme, it renders the enzyme inactive. Therefore, a competitive inhibitor often has a structural feature similar to those of the substrates whose reaction they inhibit. This means that a competitive inhibitor and enzyme substrate are in direct competition for the same binding site on a particular enzyme. Here, the competition is existing between this substrate and this competitive inhibitor for this active site. Thus, the competitive inhibitor's name. Now, for non-competitive inhibitors, let's consider this diagram. Here, when the substrate binds, in absence of an inhibitor, it forms an enzyme substrate complex. But when the inhibitor is present, it goes and binds to a different site other than the site of an active site. It binds here in a different site, inhibitor site, which changes the conformation of the enzyme structure. It also changes the conformation change, structural change in the active site. When compared to here the active site structure is changed, and thus rendering the enzyme inactive. So these are not influenced by the concentration of the substrate. There is no competition existing here between the substrate or the inhibitor. They are independent of their action. It inhibits by binding irreversibly to an enzyme, but it does not bind at an active site. It binds at its own inhibitor binding site. They also inhibit with same affinity to a free enzyme, and they also bind to the enzyme substrate complex with exact same affinity as they used to bind to the free enzyme. It can change the shape of the enzyme as well as the active site. Now coming to the last factor, that is irreversible inhibition. Now let us look at this diagram of irreversible inhibition. In irreversible inhibition, the inhibitor binds to the enzyme irreversibly to formation of the covalent bond. Here, a covalent bond is found between this inhibitor and the enzyme with the enzyme, thus permanently inactivating the enzyme. This feature is irreversible. This feature reaction occurring here is permanent, and thus no substrate can bind to it, and thus the entire process becomes irreversible. The best example I can give here is penicillin. Penicillin is an irreversible inhibitor. It works by binding to transpeptidase and inhibiting the activity of transpeptidase. Transpeptidase is an enzyme responsible for formation of bacterial cell walls. And when this enzyme is absent, the cell wall allows water to get into the bacterial cell wall, causing it to swell, burst, and die. Similarly, the other example includes aspirin and nerve gas. Now, here ends the topic regarding the factors affecting the enzyme specific activity. So, from this topic, what all questions can we expect are given below, like explain the factors affecting enzyme activity, the effect of pH on enzyme activity, the effect of temperature on enzyme activity, the effect of substrate concentration as well as enzyme concentration on enzyme activity, and types of inhibition on enzyme activity, or the types of inhibitors, or just inhibitors. So I hope students you will be able to answer all these questions after this class. I hope you understood my class. Thank you students for listening to my class. I hope I was able to give you an easy explanation regarding this topic which could be helpful for you in your examinations. In further classes, I'll be dealing with the enzyme regulation mechanism, the mechanism which is involved in enzyme regulations, that is feedback inhibition, Lac-Operon model, precursor activation, catabolic repression, etc. I hope to see you soon. Thank you. Thank you one and all.